Our second speaker is Loren Frank from UCSF. Loren is a Howard Hughes Medical Institute investigator, and one of the reasons I invited Loren was that he has a collaboration with engineers at UT Austin in developing new electrode technology, which I think you're going to hear about.
Thank you, Joe. It's a real pleasure to be here, and I want to say I come at this question of tools. As you heard, tools obviously enable our science, enable us to discover things, enable patient treatment and so on. I come at this from the perspective of trying to understand how the brain works, and so I wanted to give you a perspective on the questions that specifically motivated us to do the work that we're doing. So I like to start with this clip from the movie Ratatouille, if you don't recognize it. So this is the food critic tasting the restaurant's ratatouille, and he has this sensory experience. Wow, okay, that was profound. That drives this amazing mental time travel back to his past, right? So he remembers coming in from outside, he remembers his mother feeding him, taking care of him, the way his kitchen looked. And all of these experiences come back to him, and they do so in a flash. And this is something that I find just fascinating and amazing. The fact that all of us throughout our lives can create memories of our experiences. I would point out that the reason you are all here is in large part to create memories. And the quality of our presentation to you today will be judged in part on how well we help your brains create memories of this event, right? Because if you leave here remembering nothing, we have done a terrible job. So how does this work? Well, roughly speaking, sort of a 10,000-foot model of this is something like this. Let's suppose that you wanted to create a memory of today's session. So you're seeing and hearing and hopefully not smelling, but nonetheless, taking in sensory information from your experiences. We know that this information is processed in many stages of the brain's hierarchy and eventually is sent to specialized cortical areas that extract specific features and do other computations. Now, these areas change with experience. That is, they're plastic, the connections between the nerve cells, the synapses are dynamic, but those synapses don't seem to change quickly enough to form memories for the events of daily life. Now, this is a really good thing. You don't want to, for example, look at a chair and have your brain completely rewire itself, so the next time you looked at it, it looked completely different. So we need a lot of stability in parts of our brain so that the world seems similar time after time. But at the same time, if you want to create memories for the events of daily life and then use them, you need an area that can change very quickly. And here, then, we sort of complement this slow plasticity with another brain area, the hippocampus, which receives process information from all over the brain. And it is a site of extraordinarily rapid plasticity. So the synapses there can change very quickly. And we know that this makes it possible for us to form memories of the events of daily life, memories like those that we hope to be helping your brains form right now. So this is a process we often refer to as encoding. It involves distributed changes in synapses, certainly within the hippocampus, but likely also between the hippocampus and other brain areas. And again, I just want to make it clear, this is remarkable. The fact that we can come up with, build memories of the sights and sounds and smells and so on of experience all at once as they flow into our brains is truly amazing. But of course, the purpose of that is not just to record that information. The purpose is to be able to use it later, right? So if I were to say to you, or let's imagine a colleague says to you next week, so how was the TAMEST meeting? What did you learn? Well, what's going to happen in your brain at that time? Well, we have a sensory input, in this case, auditory information coming in. That's sent to auditory cortex, among other places, processed by many stages. That pattern of activity is changed into interpretable speech, which then goes to the hippocampus. And then the hippocampus helps orchestrate the retrieval of representations, patterns of brain activity related to the sights and the sounds and the smells and everything else that you experience, so you can recreate what this room was like. You can recreate elements of the conversations and the experiences. So this is a very distributed phenomenon, and this is the point I want to emphasize. Everything the brain does involves interactions across many, many brain areas, neurons, across tens or hundreds perhaps of regions talking to each other, and this is something that we really like to understand. I also want to say just that these circuits are involved more than just memory. There's actually good reason to believe that these same circuits are actually critical for our ability to imagine things. So if we think of ourselves as a scientist, for example, I think about what experiment to do next. And I do so by imagining things I could do and evaluating them internally. And these same circuits seem to be critical for that. Furthermore, these circuits are also implicated in a wide variety of diseases, schizophrenia, depression, PTSD, and many others. And so what we'd really like to do then is understand how in the world this works. How does the brain do this? And then how does that go wrong in cases like disease? So in my lab, we focus on this. Oops, the video has started. So this is recordings. Actually, let's see if I can go back and pause it for a moment. So we do this at the level of patterns of activity in neural circuits. So we're interested in how the spiking activity of neurons represents and transmits information to downstream areas, and how areas work together to make these memories, to retrieve them, to use them. So we work in rats. That's our model system. And so this is an example. You see a top-down recording on this side of a track that the animal is on, and we're recording, in this case, using older technology from neurons in the hippocampus, which seem to give us a sense, or as I'll show you, of what the animal is doing, where it is right now. So as many of you probably know, the hippocampus has what are called place cells. So as the animal turns around here, we've organized this. Oh, shoot, wrong button. Let's try this again. Okay, one more time. Okay, so as the animal turns around, what you'll see is individual neurons that are active where it is now, and then it starts moving, and a different set of neurons comes on, and we get this, what I think of as a quite beautiful sequence of neuronal activity, which each neuron active in a place. That neural activity continues as the animal stops, indicating where it is. Okay, and then it's, sorry, for some reason the video is cut short, but anyhow, this gives you a sense then that we can record activity in these individual regions related to ongoing experience. The end of the video, which is cut off for reasons I don't understand, shows that we also get individual events where these cells are reactivated. So patterns of brain activity related to past experience are turned back on again. This happens over the course of about 1 to 200 milliseconds. And we find that frequently the brain is reactivating these experiences from the past. This happens during what are called sharp wave ripple events, and I'll talk about them briefly again. But what I want to make a point is here is that with the technology we had, we can get this sort of picture for one brain region. But as I argued before, memories, experiences, imagination, this all involves massively distributed networks. And we just didn't have the tools to study those networks, to understand how this activity translates into activity in many downstream regions, how they work together to actually accomplish things. All right, so this leads us to the things that motivated the work we did, the set of challenges and requirements to answer the questions that we wanted to answer. So first is that the brain is really fast. So the time constants of the synapses that we're looking at are milliseconds. These patterns of reactivated brain activity happen on the time courses of 5, 10, 15 millisecond chunks of time. And so we really feel like for our questions, we need millisecond timescale resolution. Second, as I mentioned, this all depends on distributed networks, and we need to be able to measure those networks and scale them so we can put our tools where we want them anywhere in the brain. Third, computations, that is what the brain does, information processing, memory creation, engages lots of individual single neurons. And so we need to be able to measure those single neurons individually and understand how they work together. And then finally, and this relates to the points made about longevity, is activity patterns in our brain are dynamic on timescales from milliseconds to years. Right? And this is actually a huge part of the challenge, is that our brains are always changing, right? If you are creating memories, you are changing your brain by definition. And we really would like to understand that, and we'd like tools that can help us follow and measure those changes. So we really need long-lasting, stable recordings so we can watch these systems change over time. Okay, so what we did then was to team up with groups of engineers, initially at Lawrence Livermore National Labs, and as Joe mentioned, more recently also with a group at UT Austin, as well as a company called Spike Gadgets, actually run by my first graduate student, to build a system to help us get closer to understanding these circuits. So this system starts with these polymer electrodes, and I'm sorry, I can only point on one side, so I'm being left-hand biased for the room. This shows the entire electrode here. This is an example of the tip down there, and this is a zoom in. I guess I can flip sides to be fair. So the point of these is these provide individual recording contacts of a few hundred microns, many of them that can be put in the brain, and they're made of polymer, so they're soft and flexible. And one of the real challenges with working with the brain is it's squishy, and it's incredibly subject to damage. I'm sure all of you are familiar with the work on football players and traumatic brain injury from the brain smashing into the inside of the skull. The brain is very easy to damage, and so one of our goals is to build tools that measure it as non-invasively as possible. And so these are soft, flexible electrodes that allow us to go in this case, hopefully minimize the amount of damage and minimize the amount of continued trauma from having this device in the brain as it moves around. So this is then coupled to electronics, which we made to be stackable so that we could put these together in any way we wanted.
And we end up with about a 1-inch cube here, which broadcasts data off the head at about 60 megabytes a second. All right, this can be put on the head of our rats, and we can run this up through a cable, and this actually allows us to record for as long as we'd like from these devices. Now, the result of this is, oh, sorry, I'm not good with this pointer. A system that could work really well if you can get it in the brain, all right? And the problem, of course, when you're dealing with flexible electronics is that they're flexible, and so first you have to get them where they want to go. And so our colleagues at Lawrence Livermore developed this system where the electrode, which is shown in front here, and this is about 14 microns thick, so it's about 1/5 the diameter of a human hair in terms of the thickness. So what we do is we attach it to a removable silicon stiffener with polyethylene glycol, which dissolves in saline. All right, let's try this again. You can put that stiffener into a substance, let the adhesive dissolve, and pull the stiffener out. All right, and one of the nice things about this technology is it's also something that we think can be translated to primates and eventually humans, is our hope, to have a fairly non-invasive, low-impact way of getting recording devices into the brain. All right, so it turns out that this works. I probably wouldn't be here if it didn't. And this is an example of the recordings we get. So we get continuous recordings of electrical potentials measured outside of our electrodes. These are individual neuron spikes, these sharp things. And this then allows us to sample activity from wherever we put these electrodes in the brain. And I just want to illustrate that with a couple of things we did to sort of show the potential of this. So what we've done here is pick out 4 different brain regions, the hippocampus, and then 4 others, medial frontal, orbitofrontal, and ventral striatum, all of which are involved in creating memories and using those memories to make decisions in different ways and associating memory with reward. They're all in different places. They're all deep in the brain, which makes them more inaccessible to, for example, most optical imaging techniques. And so what we did is put a large number of probes across these areas. And what I'm showing you here, each little dash here is the activity of one neuron. So this is 375 simultaneously recorded neurons, all from these areas, all locked to one of those sharp wave ripple events that I mentioned earlier. These are the events from the hippocampus where we see memory replay. And I hope you can appreciate here across these areas, right at the time of the event, many of these areas just go silent for a moment with just a few neurons on. And we can quantify this on average by taking across all these neurons which ones turn down their activity and which ones turn up that activity relative to this hippocampal event. And the main point I want to make here is that we can now record an internal brain event that is something generated by the brain on its own and look at how other areas are reacting to that and understand the network and how these patterns emerge. And we think actually get some insight into how these distributed representations associated with memories are actually formed. All right. So that sort of covers those initial things. We can do things where we can put these in many areas. We can get high quality recordings. But what about that last point? How long do these last? And so we were very pleasantly surprised, and honestly, we were surprised at this, to find that they kind of last about forever. Not quite forever, but for our rats, close to forever. So what we're showing here is across a little over 5 months, the yield, that is how many of these single neurons, or single units we call them, we can record from these devices over time, either per 16-channel shank or per electrode. And you can see after some initial wound healing, we assume, there's a fairly stable and perhaps a very slight decline, but here even out to 5.5 months. And I'll just say we were not able to get recordings of this longevity before. We've taken this in a couple of animals out to 11 months, which is about half of their lifetime. And indeed, we can still record neural activity after that point. So this opens up, if we think about longitudinal studies about how the brain changes, we can now do this in individual animals in a way that we just could not before. Moreover, we can do this within a single electrode. We can follow the same neurons 24 hours a day, 7 days a week. So we did this by recording from 3 animals. We ended up with 2,232 of these single neurons, each recorded for at least 24 hours. And again, I'll say before this, in my lab, we had no 24-hour data at all. All right, and this is enough that we could probably analyze for the next 10 years and do nothing else because the richness and complexity of these data is truly extraordinary. So this is an example of one probe, one 16-channel shank, where we're looking at how long we could follow each unit. So this is over 240 hours, so 10 days. You can see we get different units on different days, but many of them we can follow. And this is across 3 animals, what proportion of these units could we follow for different lengths of time. And you see at the end, what we end up with is about 21% of them where we can follow it for over a week. All right, and so questions like, how does the brain change after an experience? How does sleep reorganize memories? Which honestly have been things we've had a hard time addressing until now, suddenly are within reach. And so we're really excited about that potential. Finally, we're not done because this is just the first step. So we're now collaborating with colleagues, including Peter Dennis, who's an extraordinary engineer at Lawrence Berkeley National Labs. He's built this e-chip, which is a 1944 channel device in 5mm square for amplifying brain signals. We're actually reducing the density to 528 channels, adding digitization, and then also coupling these directly to the probes. It turns out that coupling silicon to soft, flexible things is not trivial. This is a hard problem, but it's something that we're really excited to be working on and think we can solve. And then we're coupling it, as mentioned, both to probes from our current collaborators at Lawrence Livermore, but also our newer colleagues at UT Austin, Cheung and Lan Luan, who have built these incredible probes that are 1 micron thick. So they're about the sort of stiffness of a cell membrane, and they have the potential to integrate. In fact, they do seem to integrate seamlessly into brain tissue. So we're really excited about the potential there. And then the goal is, again, to have individual devices and up to about 16 of them, which would be around 8,000 electrodes, that we can put in different brain areas to measure activity across the lifetime of the animal at millisecond resolution. And if any of you have any really large hard drives, please let me know. All right, so in conclusion, we're really excited about this system because it solved a number of the problems that we had in our field we had. It allows us to do large scale, so in this case, 1,000 electrode, 1024 channel recordings. We can distribute these recordings where we want to. And again, I want to emphasize that the brain works through distributed computation. And we feel like our first step in understanding that is being able to measure that distributed activity, both in health and in disease. These also give us very long-lasting stable recordings, which we're excited about. We have then, as I mentioned, a path towards another 8-fold increase in density. And the most important thing, right? These tools are helping us ask new questions. So I just want to acknowledge, this is, as mentioned before, like at the Allen Institute, this stuff happens when you have a great team. So I've been fortunate to have wonderful people in my lab. I want to highlight Jason Cheung, who's now back in medical school, who really pioneered this and made it work. I'll just mention that he did a lot of this, slightly over time. He did this because he wants to be a neurosurgeon, and he developed a lot of the surgical techniques to be compatible with human work. And then finally, our colleagues, again, at Lawrence Livermore, at UT Austin, Lawrence Berkeley, that really make this happen. Thanks.
Thanks, Loren. We have a few minutes for questions.
Very interesting. My question is, I realize you're sort of at an initial state on a lot of this. Is the technology, can you take that technology and use it in like an operant chamber, Skinner box kind of?
Absolutely, yeah.
Okay.
So we have these, I didn't show this, but in fact, the 10-day data was, in our case, an animal running about 750 meters to a kilometer a day on a large track. And it's on a thing that allows the animal to spin around. So you can put these in a chamber and you can record continuously if you want
That'd
to.
be great, because then you can really do complex behavior.
Absolutely,
My
absolutely.
second question is, I saw your N was 3. Have you looked at all at sex differences? Again,
Yes.
recognizing that you're early on.
No, that's a great question, and we have not yet. So for the moment, we have, in a very biased way, stuck with male rats because they're twice the size of female rats.
Yeah, no.
And that makes a physical difference in terms of carrying this stuff. One of the things we're really hopeful for with the new technology is it's much smaller and lighter. So this should be able to make it easy to extend, and as part of our ongoing work, we're also trying to distribute this to labs that work with mice, songbirds, bats, rats, and primates to get this working across many species of many different sizes and both sexes.
Thanks.
Thank you, Roderick Pettigrew. Fascinating. My question pertains to concerns about sampling. With the electrodes, 1024 is a lot of electrodes given a small animal, but they are physical structures entering the brain. You run up against the Heisenberg uncertainty principle. Just so concerned about that. And a bit previewing perhaps what Mark Schnitzer will talk about, a complementary technology where he uses fluorescence to do a similar sort of thing with the microendoscope. I may be giving your talk away, Mark. Maybe not going to talk about this at all. But those techniques would seem to be complementary.
Yes. Yeah, so let me start with the first comment. One of the most amazing things about the brain is that you can understand anything at all from recording the activity of a small number of neurons, which seems slightly absurd, right? In our rat brains, we have 100 to 200 million neurons. And the idea that you could make any sense out of anything by recording a small number of them seems strange. And yet it is true that, for example, as I showed you in that video, we can relate the activity of our hippocampal neurons extraordinarily well to where the animal is in space, and even to, as far as we can tell, things that it's thinking about. So the suggestion there is that there is at some level a profound degree of redundancy in the system, and that redundancy makes it possible for us to sample small numbers of neurons. But I just want to highlight your point. We're at 10 to the 3 now, all right? We need to be at 10 to the 8 for rats. We need to be at 10 to the 10 for humans, right? There is a big gap there. The second point is I by no means think that everyone should do this, right? There are many complementary technologies, and indeed part of our current distribution grant is to try and figure out how we can couple these devices and their millisecond time resolution to optical methods, which have the ability to do cell type specificity and fluorescence measurements, but are more limited in time, sort of the time resolution of their measurements. So that's something we're actively working on.
Okay, thank you very much, Loren.